let's see another example where NGS can be used. This is forensic science. Nobody uses forensic science, but we used to see very lot of series about that. So what happens is you go to a spot, you collect evidence, you collect spots of blood, you collect anything, even the hair. If you get 2 or 3 hair strand, you can extract DNA out of it if they have the bulb intact. So what can be done in forensic science field using those samples? You again extract DNA, sequence it, genotype it to find out if you have the database, that if you have so if you have suspects, you can sequence their DNA also, you can sequence the evidence DNA also, and you try to find out who might be the culprit. So that is one thing. Then if you don't have suspect leads, you can just investigate the leads. Let's say one of the suspect is a family from the original criminal, but you don't know that. When you sequence and this is a very famous case, the DNA was sequenced from the blood samples, and when matched with 10 or 15 suspects, they found one of the suspect whose genome was matching with the evidence, not fully, but it was around it. I am not sure about the number exactly, but it was uh, very nearby to that evidence. Suspecting, so the investigator, they finalized that the DNA belonged to someone in the family of that suspect. It's not of suspect, that's for sure, but someone in the family. And that they again tracked all the family members and tried to caught the criminal. So this is another exciting field if you are excited about solving all these cases. Again, what I said, you can get any kind of stain or spot in the crime scene, and you can just collect it and try to genotype it. You can get a mix of blood. It could be animal blood and human blood mix, but when you genotype it, you can actually tell the differences in the blood sample, and it will help our investigation officers to close the case very really quickly. Again, then in plant science, how will you use the NGS thing in plant science? You can just prepare the library, you can sequence it, and you can do a lot of things, right from gene expression to protein analysis. Comparative omics is like doing a lot of other stuff and then just combining all the omics stuff, transcriptomics, mironomics, epigenomics, other kind of thing. You can also find out genetic variation. Let's say there are 2 species of maize, and one is drought sensitive, another is not sensitive, but they both are maize. It means their genome should match uh, at a very high number. When you sequence both of the species, both of the samples, one is drought resistant and then other one is not, and you try to find out the difference, it might be that there are some SNPs or variations which is causing one to be a resistant plant and another one is not. So that how you can do that also. Then using NMR, you can do NMR or LCMS, you can do metabolite profiling. These are the secondary metabolites which can be used for lot of things, maybe for medicinal use also. Then plant pathogen interaction using microbiome or transcriptome or other things. Everything requires bioinformatic analysis. So any of the data you will get, you will do bioinformatic analysis, you will again prepare the report, you will talk to your colleague, the researcher. On the basis of your report, they will go ahead and modify the genomes of that plant. Let's say you got a variant which is making one sample of a maize drought resistant, but other is not. You told that to your colleague, your researcher, and they came up with a strategy. Let's say using CRISPR-Cas, they modify the part in other maize also, and they will try to see if modifying that part or introducing that mutation in the other plant, will that plant will become drought resistant, stress resistant or not. So this is just one example, but there are a lot of things you can do in plant biology also. You can improve the quality of grains. which is you can improve the number of the quantity of grains. So there are a lot of possibilities. You can identify the disease again. You can identify the disease also in the plants. If the plant crop is not doing well, you can just try to find out what could be the cause. So there are a lot of possibility in this field also.